Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
We often focus on the what in cancer, right? The specific mutations causing problems. But today we're digging into a Nature paper that asks something different. Why there?
Yeah.
Why does one cell turn cancerous while its neighbor with the same bad mutation just doesn't? And the answer they found is pretty surprising. It's not another gene. It's timing, speed. They call it total cell cycle duration, or TCA.
Yeah, it tackles this really fascinating puzzle. We know that potentially cancer-causing mutations, like losing the ARB gene, are actually pretty common in perfectly normal tissues. Most cells with these hits don't become cancer. They don't necessarily die or get zapped by the immune system. They often just sort of stop. They exit the cycle, mimicking normal development.
Right. So the classic hallmarks of cancer describe the end state, the uncontrolled growth, avoiding death signals. But they don't really explain why this cell lineage is vulnerable and that one isn't, even with the same starting mutation. This study seems to point upstream to the cell's own internal clock.
Exactly. They zoomed in on a specific control system, the SKP2, P27, CDK1, CDK2 axis. Think of P27 as a brake on the cell cycle holding it before division. SKP2. That's the molecule that basically removes the brake. So by tweaking this system, you directly control how fast the cell cycle runs the TP.
Okay, so they identified this control knob. The big question then is, can manipulating it actually stop a tumor from forming? They looked at retinoblastoma using mice that were missing both ARB and P107 genes, which normally get these eye tumors.
And their approach was, well, elegantly simple in concept. They just reduced the amount of that accelerator molecule, SKT2. They engineered mice with only one functional copy of the SKP2 gene instead of two, so roughly half the SKP2.
And the outcome?
Stunning, really. Just reducing SKP2 by half completely stopped the retinoblastomas. 0 tumors in 40 eyes tested in those SKP2 heterozygous mice.
Wait, 0. So you're saying these cells, all tried for cancer, didn't need to be killed off. They just needed to slow down.
Precisely. And this is the crucial part. This tumor suppression happened without the usual suspects. No increase in cell death, no jump in senescence, no evidence of immune cleanup, didn't even fix the underlying DNA damage issues. It suggests TLC is this separate upstream dial you can turn, independent of those other hallmarks.
That's pretty remarkable. Stopping cancer without killing cells. So if slowing down works, is that speed difference already there before cancer? Like, does TLC naturally vary between cell types that are prone to cancer versus those that resist it?
That's what they tested next in Figure 4. They used a clever technique, staggered EDU, BRDU double labeling. It lets you measure the total cell cycle time, DSC, and also the S phase duration, ETs, for specific cell types within the retina.
Okay, so let's pause for a thought experiment here for you listening. Imagine you have one cell type that races through its cycle in, say, 26 hours, and another, more resistant type, takes a leisurely 143 hours. Which one seems more likely to rack up enough errors to actually become cancerous?
Well, it's the faster one, right? And that's exactly what they saw. The amacrine cells, which are the ones prone to becoming retinoblastoma in this model, had a TC of just 26 hours, really fast. Compare that to the resistant Miller cells, 143 hours, or even the horizontal cells at 77 hours. It's a huge difference. It's almost like being slow is, in itself, a tumor suppressor mechanism.
And crucially, when they introduced those tumor-suppressing genetic changes, like having only one copy of SKP2, what happened to TC?
It confirmed the link. Every change that stopped tumors also significantly increased the TC of those fast amacrine cells. For instance, the SKP2 reduction pushed their TC from 26 hours up to 43 hours. Slowing them down prevented the transformation.
Okay, that's compelling for retinoblastoma. But is this a general rule or just specific to that RB pathway in the eye?
That's the question Figure 5 addresses. They looked across different tissues, pituitary, lung, and different cancer-driving mutations.
And the finding holds.
It does. The cell lineage with the shortest TC was consistently the one that gave rise to the tumor. Didn't matter if it was retina, pituitary, or lung. Didn't matter if the driver was RB loss or Kras activation or Braf. The speed rule applied.
Can you give an example from the lung model?
Sure. In a model, Driven by losing both RB and P53, the susceptible cells were neuroendocrine cells, their TC. Just 11 hours, extremely fast. Compare that to nearby resistant club cells taking 42 hours.
Wow, 11 hours versus 42. That's a massive difference in opportunity for errors.
Exactly. And even if you switch the driver completely, say to an activating Kras mutation, the rule still works. In that context, the susceptible cell type shifts, it becomes the AT2 pneumocytes. But guess what? They now have the shortest TC in that context, around 21 hours. The kinetic vulnerability follows the cell of origin.
So the big innovation here seems to be shifting our view of cancer risk. It's not purely about the static gene mutations, but also about these dynamic physiological properties like cell cycle speed.
Precisely. TAA acts like this foundational modifier. It's a distinct knob that can be turned separate from fixing the initiating mutation itself.
And the implication for you, thinking about future therapies, is potentially quite profound. Maybe we don't always need harsh chemo to kill initiated cells. Could we develop gentler, maybe intermittent treatments designed just to slow down those high-risk clones?
Right, things that might stabilize P27 that break or inhibit SKP2 or the CDKs slightly. Basically, nudge them towards exiting the cycle, making them behave more like those naturally resistant cells, a chemo prevention strategy based on kinetics.
Okay, so to wrap up the key takeaways for you. 1. The shortest total cell cycle duration, TDA, seems to pinpoint the cancer-susceptible cell lineage within a tissue. 2. Tumor suppression, at least in these models, works by fundamentally lengthening that TC in the originating cells. And 3, this speed principle appears pretty general, holding up across different tissues and driver mutations.
There is a really interesting limitation, though, a bit of a puzzle they raise. While relative speed matters within a context, the absolute speed limit isn't universal. Remember, a TC of 21 hours was fast and cancer-prone with Kras, but a very similar TC, 22 hours, was actually resistant in a different lung model with RBP53 loss. So the threshold for too fast seems to depend on the specific initiating mutation. Why is that? What sets that critical speed limit differently in each case?
So the context of the initial hit changes the definition of dangerously fast. That's definitely something to chew on.